Okay, this video I'm going to talk about the essential structure of a bacterial gene. So what I'm first going to do is I'm just going to draw a line here, and this is to represent the actual gene region. And this is the 5' end, and this is the 3' end of the gene. Now, essentially, transcription, if it's going to start here, will have two sites upstream, and this is upstream, one that we'll call the -10 site and one that we'll call the -35 site. And effectively these are two very, very specific sequences of DNA. The -10 region has what we call a consensus sequence of T, A, T, A, A, T. And the -35 site will have a consensus sequence of T, T, G, A, C, A. Now what these two sites together constitute is a promoter element. And the promoter element is recognized by the RNA polymerase, and specifically it's recognized by the sigma factor. Now, when the RNA polymerase recognizes this promoter element, it knows that it's going to begin transcription downstream of this element, about 10 base pairs downstream of the -10 region. And the RNA polymerase then starts making RNA. The RNA polymerase is a large molecule. It's about approximately 500,000 Daltons in size, and it consists of 4 different subunits. These are 2 alpha subunits, we write it like this, a beta and a beta prime subunit. The 5th subunit of RNA polymerase, the sigma factor, that's sigma. The sigma factor is used in recognizing this promoter region, but afterwards it falls off, it's no longer needed. These 4 subunits, which are 4 proteins forming what we call a tetramer, these 4 subunits together constitute the RNA polymerase, and they start making the RNA. So this is what's needed for initiation of transcription. Transcription then proceeds. The messenger RNA is being produced, and it continues to be produced with the successive addition of new nucleotides. And these are RNA nucleotides, until we get close to the end of the gene. Now, what we're going to see at the end of the gene is a particular sequence that's in the DNA. And there are 2 kinds of transcription termination. One is row independent, and one is row dependent. So for row independent termination, we'll take that first. You've got, first of all, a sequence in the DNA that when it is transcribed into RNA, it forms this stem and loop structure. It's what we call having dyad symmetry. And afterwards, there's a run of nucleotides that are all A nucleotides. And what happens is that the A nucleotides bind very weakly to the U nucleotides on the RNA, and as a consequence, this dyad structure, this stem and loop structure, followed by this run of weak bases, causes the RNA to dissociate from the DNA. And so this is row-independent termination, and row-independent termination is caused by features of the DNA sequence. Alternatively, row-dependent termination is caused by an additional protein that is employed, and this protein binds to the messenger RNA itself, and it causes the whole complex to destabilize. So row-dependent termination is caused by a specific protein. So this is transcription initiation. This is the generation of the messenger RNA. So this is elongation, and this here is termination. The result is a messenger RNA that extends from the 5' region to the 3' region, and it corresponds to the transcription start site all the way down to the termination site. Within this messenger RNA, somewhere along here, we will have the start codon and the stop codon, which are used for translation, but I think that's for an entirely different video.